Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome to The Explainer. Today, we're diving into a medical revolution that's happening right now. The ability to edit our own genetic code, not in some distant lab, but directly inside our bodies to cure diseases that we once thought were completely incurable.
I mean, think about it. What if we could stop just treating symptoms? This is a totally different way of thinking about medicine. It's an idea that's leaping from the pages of science fiction into actual clinical reality, fixing the problem where it starts.
And how do we do that? By correcting the tiny, single-letter mistakes, the typos, in our DNA. For decades, this has been the Holy Grail. We're talking about a fundamental shift, aiming for a one-time fix, a permanent cure, by going right to the root of the problem.
So how do you actually pull this off? Well, scientists have come up with two really powerful but also really different strategies for how to fix our genetic code right inside the body.
Okay, so the key difference is all about the approach. Gene replacement, think of it like adding a brand new, perfect cookbook to your kitchen. You don't throw out the old, smudged one, you just leave it on the shelf and use the new one instead. Now, gene editing is totally different. That's like taking a pen, finding the exact typo in that original cookbook and correcting it right on the page. One is an add-on, the other is a direct fix.
But here's the billion-dollar question. How do you get these new genetic instructions into trillions of cells scattered all over a person's body? Well, for that first wave of therapies, the answer came from a tiny, re-engineered virus. It's called an adeno-associated virus, or AAV for short.
And here's why these AAVs are so brilliant for gene replacement. They don't go messing around with our main chromosomes. Instead, the new gene they deliver forms its own stable, independent little circle of DNA called an episome. Think of it like a durable instruction manual that just hangs out in the cell's nucleus, turning that cell into a non-stop factory for the exact protein it was missing. It's incredibly clever.
Of course, it's not perfect. There are a couple of major catches. First, the AAV delivery truck is tiny. It's got a really small cargo bed, which means you can only fit small genes inside. Second, and this is a big one, a huge chunk of the population, up to 70% of us, has already been exposed to this virus. Our immune systems recognize it and will just shut the therapy down before it even starts. These two flaws literally shape the entire first generation of genetic medicines.
So, with these huge hurdles, what did scientists do? Well, they got creative. Let's take a look at how they brilliantly navigated these problems to create 4 landmark therapies that are now approved. Each one is a story of escalating ambition.
You can just see the story of progress right here in this table. Luxturna started smart. It targeted the eye, which is immune-privileged, basically a place the immune system doesn't patrol. Zolgensma went bigger, using a special AAV that could sneak across the blood-brain barrier to treat a neurological disease. Then you have Elivirus, which tackled that cargo limit head-on by designing a mini but still functional version of a huge gene. And finally, Bicuvez showed us you can actually optimize the payload itself, using a super potent gene to get way more bang for your buck.
Gene replacement was a total game changer, no doubt. But the next step? Well, it's even more profound. We're talking about directly editing our original genetic source code, and for that, you need a completely new set of tools.
And that's where CRISPR comes in. You see, the early gene editing tools, ZFNs and TALENs, they were really clunky. They relied on custom building these complex proteins, which was super difficult and time-consuming. But CRISPR, it uses a simple, programmable piece of RNA as its guide. This was the breakthrough. It made gene editing fast, cheap, and easy enough for basically any lab in the world to do. It completely democratized the whole field.
So after CRISPR makes its cut, the cell's own repair crews get to work. And what's cool is that we can actually take advantage of their different work styles.
If we just want to shut a gene off, we rely on the fast and messy crew, NHEJ, which usually makes a small error that breaks the gene. Perfect. But if we want to make a precise correction, we give the cell a correct template for the precision architect crew, HDR, to follow, ensuring a perfect fix.
All right, so CRISPR is this incredible tool, but delivering it creates a whole new challenge. To safely edit our DNA inside the body, we really needed a different kind of delivery truck. AAV, our old workhorse, just wasn't the right tool for this new, super precise job.
And this quote, it just nails the problem perfectly. With gene replacement, you want that protein factory running 24/7, maybe for years. But with gene editing, you've got these molecular scissors. You want them to get in, make one specific cut, and then get out. Leaving them active in the cell forever would be way too risky. They might eventually cut somewhere they're not supposed to.
This is exactly why the field pivoted to lipid nanoparticles, or LNPs. They are perfect for this hit-and-run delivery. They carry temporary mRNA instructions. The cell uses these instructions to build the CRISPR editor, but then the mRNA just degrades and disappears within a few days. This temporary approach is a massive safety upgrade, and it's perfectly tailored for making a permanent change to our DNA.
So all this technology is bringing us into an absolutely incredible new era of medicine. But with this kind of power comes, well, profound responsibility. We're now being forced to confront some of the biggest questions of our time.
And this is where we have to draw a very bright, very clear ethical line. All the therapies we're talking about today fall under somatic editing. That means you're treating the disease in one person's body, and those changes are not passed on to their children. The other side of that line is germline editing, changing sperm, eggs, or embryos. That kind of change would alter the human gene pool forever, and right now there's a broad international consensus that we should not be doing that in humans.
So what's the bottom line here? First, we are living through a truly transformative time. We have approved therapies that add genes, and a whole new wave is coming that will edit them. Second, our tools are getting smarter. We're moving from the AAV workhorse to more specialized tools like LNPs. And finally, the biggest hurdles ahead might not even be scientific. They're about safety, the incredibly high cost, and making sure everyone who needs these cures can actually get them.
And that's really where we're left. We now have the power to edit the very code of life. The question isn't if we can do it anymore, we can. The real question, the one that's going to define not just the future of medicine, but maybe what it means to be human, is how will we choose to use this power?
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.